temperate lakes. Susie. I don't know how to work this. There we go. You do it.
I'm all right with that. Yep, cool. Everyone can hear me? Yep, thanks very much everyone for coming along and for the organisers for organising this workshop again. It's great to be back here and see so many familiar faces and always a bunch of new faces, and it's always really neat for us to come over here and hear about what's happening in Australia. So today my talk's really about looking back into the past and thinking about how we might use some historical data to better guide the future. So New Zealand has over 3,800 lakes, greater than 1 hectare in size, and the location of those blue dots shows where each of those lakes is. So our lakes are diverse in every way imaginable. So our largest lake, Lake Taupo, is over 600 square kilometres. Our deepest lake, Lake Karoko, is over 460 metres deep. And then we have many shallow alpine lakes, large coastal lakes, and a huge diversity of different types of lakes. So around about a third of our lowland lakes experience cyanobacterial blooms, but also our oligotrophic lakes have really high abundances of cyanobacteria. So these are really poorly studied, but these lakes have huge abundance of picocyanobacteria. And until recently, I guess the methods have prevented us understanding these picocyanobacteria diversity and actually what their functioning is. But they're really, really important parts of these communities. So when we think back to these shallow lowland lakes, which are experiencing cyanobacterial blooms, most of us are aware that increase in nutrients drives cyanobacterial blooms. But one of the other questions that we might ask is, in some of our systems, our shallow lake systems, have these cyanobacterial blooms always been there? Have they been a natural occurrence? Or are there any other mechanisms that might be causing these cyanobacterial blooms? And likewise, in these oligotrophic lakes, we know nothing about the historic picocyanobacterial communities or any indications of whether they're changing and whether some of those changes, if we were able to monitor and understand them, might show us whether there have been changes in water quality and they might be approaching tipping points. So to look back at historical cyanobacterial communities, this project we've worked on 6 different lakes in New Zealand. These lakes are very different, some large lakes, some large shallow lakes, some quite deep, very beautiful lakes. And for the purpose of time today, I'm just going to focus on 3 lakes. So Lake Paunui, Lake Naroto and Lake Paringa. And as I go through, I'm going to introduce each of those lakes to you. Okay, so the technique that we've been using is using paleolimnology. So year by year, sediment is laid down into the bottom of a lake. And if we take a sediment core from that lake and divide it into layers and date those layers, we can learn about the past historical communities in those lakes, what's happened in the catchment around them, and about the water quality. So paleolimnology is primarily, if we're looking at biological communities, is primarily relied on organisms that leave fossils or remains, such as diatoms, in the sediment cores. But the advent of molecular technologies is now allowing us to look at a whole suite of new organisms, soft-bodied organisms, one of those being cyanobacteria. Right. So what I want to do now is just give you these 3 different case studies of these lakes. They're all in slightly different stages of analysis, but it'll give you an indication of the different types of techniques we're using and just the different types of information that we can gather from this approach. So the laboratory methods that we've been using, they don't need any introduction. People have already talked about them, both high throughput sequencing, metabarcoding, initially targeting cyanobacterial communities, but also I'm going to present a little bit of data on entire bacterial communities, and then quantitative PCR, and I'm going to present some data targeting the microcystin, one of the microcystin genes, and also total cyanobacterial concentrations. So the first lake is Lake Paunui. It's a wonderful little lake at the bottom of the North Island of New Zealand. One of the things that makes it quite unique in the North Island of New Zealand is that it's one of the only lowland lakes where about 95% of its vegetation is still intact. So all of the tributaries that run into this lake come through this very pristine forest here. It's traditionally been thought of as a clear water lake, and it has some quite diverse fish communities and macrophyte communities. But in the last 5 or 10 years, we've seen the formation of cyanobacterial blooms, such as this one, from spring through to autumn. And so in the absence of land use change, we asked ourselves, what might be the cause of this cyanobacterial bloom? Has it always been there, but we're only now starting to monitor regularly enough that we're picking it up, or has there been some other change? Okay, so if we just look at this is the data from the sediment core. So just to orientate you, I've flipped the sediment core on its side here, and this is now the most recent surface, going back 1,500 years. So we took a 1.5 metre sediment core, which represents 1,500 years of lake history. So the top graph there shows the cyanobacterial community composition generated from high throughput sequencing data, and the bottom graph is qPCR data of 16S copy numbers. So we're trying to layer various different techniques on top of each other just to kind of provide more evidence, I guess, that these changes that we're seeing are real. And we also got a whole lot of pigment data and other things that I won't show today. So what we see from this data is that cyanobacteria have always been present in this lake, but back around about 700 or 800 years ago, these were all picocyanobacteria, so cyanobacteria that we might associate with more oligotrophic systems. Around about the time Europeans arrived, we started to see the presence of bloom forming, more species that we much more associate with bloom formation, such as Dolichospermum is the dominant one in this lake. So that's what's happening in terms of the cyanobacteria, but what might have been the cause of these blooms. So we've got lots of different evidence, but I'm just wanting to show you some of the metabarcoding, 16S bacterial metabarcoding data. So again, just to orientate you, I've laid the sediment core on its side. This is the surface, the most recent sediment going back 1,500 years. And in this catchment, Maori's or Polynesian settlement was around about 700 years ago. And each of those different coloured bars on there represents a different type of bacteria phyla. So what we can see from looking at here from about 700 years ago to 1,500 years ago, the bacterial community was reasonably stable. After Polynesian arrival, there is some change in the community through this region here. And although they didn't burn the catchment of Lake Pounui, there was a lot of burning and habitat modification that went on in the much larger region. But what hopefully is very apparent from this graph is there's this real dramatic change in the microbial community at around about 1870. So any ideas what might have caused this? And Michelle can't answer. What's that, sorry? Volcanic eruption, yep. Most of the big volcanic eruptions in New Zealand were a bit earlier than that, but yep, good idea, could be, yep. European settlement, yep. So forest burning, but they didn't happen so much in this catchment. European settlement's on the right track. Agriculture?
Yes.
Yeah, although most of this catchment, probably only 5% of it is in agriculture. So in the 1970s in New Zealand, when Europeans arrived, the British, they wanted to make New Zealand more like England, and so they brought a lot of introduced animals into New Zealand with terrible consequences. And most devastatingly, into Lake Pounui, they brought trout and probably most devastatingly redfin perch, or European perch. So they feed on zooplankton, which then selectively feed on aquatic organisms. And one of the reasons we think, it could be things like earthquakes and volcanic eruptions as well, but one of the reasons we suspect this is at least one of the main causes is this yellow bacteria that I've highlighted here. When we look at its gene sequence, it's one that is associated with the intestinal tracts of fish such as perch. So some quite good evidence that they might at least have started this, might be the cause and have started this lake off on a completely different trajectory. And if we delve down, of course, with this high throughput sequencing data, you can delve down into any kinds of detail. But just looking at things, we start to see an increase in bacteria associated with denitrification, which is a good indication that this lake is becoming more eutrophic, and we see the rise of cyanobacteria there. So this is a good example of how we might be able to use this kind of information to help us think about what the lake was once like before human settlement, what we might want to set as restoration aspirations, and how we might go about, what some of the first steps might be for restoration of this lake. Okay, so the next lake I wanted to talk about is Lake Naroto. In contrast to Lake Ponui, you'll see from this picture here, its catchment is very, very modified. So this is in the central North Island of New Zealand in a highly agricultural area. It would have been burnt with the first Polynesian arrival, but then burnt some more and introduced fish put into this lake as Europeans started to arrive. And it has quite a long history of cyanobacterial blooms, at least as long as records have been maintained. So looking at the data, this again is taking a sediment core. This one's about 800, a bit longer than, about 1,000 years back in time. And I've just highlighted there in the different colours, the different phases, European, Polynesian and pre-human, So as we see in all of our lakes, there is always cyanobacteria there, but we see some rapid increases with the arrival of Europeans. And we still need to delve down into more of this data and analyse some of the parallel data to understand a bit more about what are the key causes of this. But I wanted to use this example to show that we can also look at other genes in these sediment cores. So in this example here, we've looked for the presence of microcystin E gene, and we see that only occurs in this European phase. So we know that these lakes haven't always contained toxin-producing cyanobacteria. Great, okay, and so the last example is Lake Puringa. This is a lake situated on the west coast of the South Island. That red line there shows the Alpine Fault line, which is one of the major fault lines in New Zealand. It's the boundary of the Pacific and the Australian plates. And so this fault line here has a major magnitude 8 earthquake around about every 300 years. And so this lake here just happens to sit right on this Alpine Fault. So we thought, what a fantastic opportunity to look at how lake communities respond to natural disturbance. Most of the time we think about looking at how lakes respond to human or anthropogenic disturbances. But here's an example of a natural disturbance that occurs every 300 years, and we might just be able to have a look at how communities respond. So when we get a magnitude 8 earthquake, what happens in the lake is a huge and sudden input of sediment into the lake. Okay. So this is high throughput sequencing data targeting just cyanobacteria, and we're going back 1100 years along the bottom here. And actually in hindsight probably yellow wasn't a great colour, but all of that yellow bars there is a Cyanobacteria synovium. So if you looked at that, you might think that there hasn't actually been a lot of change in the community. And those red dashed lines there show where each of these major earthquake events has occurred. So I've got some great colleagues that work on earthquakes and characterising earthquakes, so they've spent a huge amount of effort and money in characterising the exact times of those earthquake events. So it was always Cyanobacteria dominated, and it still is Cyanobacterial dominated. But if we start to delve down into this data and look at things at a molecular species level, what we see is that there have actually been some changes occurring. So this is a multi-dimensional scaling plot. The closer the symbols, the closer the biological, or the closer the cyanobacterial communities. And essentially these triangles, squares, diamonds, stars, these are the older sediment. And it seems like that when there are these disturbance events, there is some resilience, and the community seems to be able to return back to what it was like pre-earthquake. But one of the questions we asked ourselves was, were there cumulative effects? So can it respond to a number of disturbance events, but ultimately that will be too much and it will push the community in a different direction. And it looks like this might be what this is happening, because these are the newest samples here, and you can see quite a diverse and very different community to what it was historically. So a little bit more work to go on analysing that. And this is just looking at the species richness, which shows a similar sort of pattern. So going back historically, quite a stable number of different species there, but there's now a huge change in diversity. And so this also does highlight one of the challenges with looking back in sediment cores and DNA, is that we do need to learn more about DNA degradation in these sediment cores, and we've got a number of tests and things underway at the moment just to make sure that those patterns that we're seeing are real, not just some artefact of degradation. And to finish up, just to highlight a project that we're just starting, which is a continuation of this work, which is called Our Lakes Health Past, Present and Future. So we decided that 6 lakes wasn't enough, and so we decided that we were going to collect sediment cores from, we just got funded to collect sediment cores from 380 lakes across New Zealand, so that'll be 10% of our lakes. So it's an extremely challenging project, both scientifically and logistically, but hopefully we can generate a wealth of data that helps us understand which lakes we need to prioritise for protection, and also guides restoration of lakes which are in need of help. And so if you want to come and core and visit some lakes, we will need some hands over the next few years. And thank you very much for listening, and to these organisations, or the Ministry of Business, Innovation and Employment, for funding these 2 projects.
Thank you, Susie. Oops. Your second last slide answered my question, so now that I haven't got one, I'll open it up to the audience if anybody has a question for Susie.
It's very
Well,
it's very interesting, you know. I guess it's exactly what's happening in Australia as well. We've been seeing the algal bloom more frequently than we saw some 50 years or 70 years ago. So it's quite interesting to look at our own backyard, how the European settlement and the changes contributed to the frequent occurrence of algal blooms in Murray-Darling Basin.
Thank you.
Brad?
Just
wait.
You said the cyanobacterial blooms had only really become a big problem in the last 5 years, but you then associated their ascendancy with European settlement and introduction of introduced fish species. Why did it take 210 years for people to start to notice the problem?
Yeah, really good question. So I guess the change in food web would have been, it would have been quite dramatic to start with, but then it's also probably been a slow process. But I don't think it's just that, it's about the build-up. There's been a gradual build-up of nutrients in the sediment and in the water column, and we see that. So now, for example, there's a huge amount of nutrient cycling that goes on in that lake. So I think it just started the process of creating a whole lot of nutrient feedback loops. So I think that was probably the start of triggering it and sending it off on its new trajectory toward eutrophication. But now there's a whole lot of other processes that have started on top of that. Yeah.
I have got a question. One of the things that's always intrigued me is the difference between Australia and New Zealand in the range of species that produce toxins and the range of toxins that are being produced. You seem to have a wide range of fairly exotic everything. What do you think is the main cause of the big difference between Australia and New Zealand in the species that are present and the toxins that they produce?
Well, that's a hard question, but actually I think it was really interesting to see the talk this morning where you had found cyanotoxins in the Murray-Darling, and that Cuspithrix was probably your most likely producer, because that's our cyanotoxin planktonic producer. I also know that Scytonema, I think that Barbara has saxitoxin producing Scytonema. So I think that probably once we start studying and looking more, that actually we'll find there's a lot of commonalities between our toxin-producing species. And I'm sure that you'll find cyanotoxin-producing Phimidium or Microcolus. Anabina circinalis or Dolichospermum circinale is the saxitoxin producer. We've not found that. So there are differences, but yeah, we see it worldwide, I guess. So it's not necessarily a New Zealand Australian.
Yeah, I think SA Water have found cyanotoxin producing Formidium in some of their reservoirs.
Okay.
Yeah.
Yeah.
All right, thank you, Susie.
Thank you.
Put your hands together. Thank you.